Carcinogenesis and chemoprevention. So the second half will be working on chemoprevention, and just to give the background, the chemoprevention was really came in effect in 1974-75, and around that time the term chemoprevention was coined by Michael Spohn at NCI, and 75-76, and at that time they had, like, we are reading the literature, and I was reading also about his papers in Nature and Science. They were just coming back and forth. So fortunately what happened was that I got a job and started working with Michael Spohn on his projects with my boss, and my boss was directly working with him. So I got to learn a lot about cancer prevention, and then I spent like many years in cancer prevention, discovering new drugs, identifying compounds, looking at the effects. And so I want to give you the overview of cancer prevention, and then some of there are like 2 or 3 figures in there which show the work probably that had come from our group. But then more I'll go as we go along in the afternoon session. I'll start showing my own work. So this will not have too much of my work, but chemoprevention in terms of its concepts. Okay. So when we look at the schematic diagram of a normal cell becoming cancer, so you have a normal cell. We just talked about normal cells. The normal cells get insult of a carcinogen, and the cell becomes initiated. So from this stage to this stage is called the initiation, and then the initiated cells, as we talked about the promoters, so they get promotion after using the effect of all the exogenous factors and endogenous factors like hormones, exogenous factors like TPA for skin cancer that we were talking about. And so you have a promotion occurring, and then after promotion the cells become into the next phase called progression, because this now promoted cell, the clone that is created, starts growing faster, and it's called progression. And once the progression occurs, some of them are going to survive, and then some of it is going to be death, because it's a mortality. So if you use this chart from normal to cancer cell and survival and motility, you have a initiation occurring. So any drug that you put before initiation has occurred will be anti-initiating drugs. So cancer prevention is either anti-initiating drugs or anti-promoting drugs. And the anti-initiating drugs are mostly like antioxidants, and they have properties like that. And so they prevent, they either induce the glutathione transferase, or they prevent the transfer of the cells by inducing, by inhibiting the phase 1 enzymes and things like that. So anti-initiating agents are given before the initiation. Anti-promotional agents are given after the carcinogen is given. So in experimental model, you give a carcinogen like MNU induces mammary cancers with a single injection. So you give a single injection, you wait for few days. Usually we start after a week of the carcinogen treatment, and then the promotion is going to start. So at that time, whatever compound we give is called anti-promotional compound, right? And so we have 4 groups. We have control, we have drug that we give as an anti-initiation. So we start 1 week before the carcinogen injection, and we stop 1 week after the carcinogen injection. That's anti-initiation. So like, the carcinogen is given in between. So here you have carcinogen, we start the anti-initiating drug here, you give carcinogen, you stop the treatment with the drug, and then if you see an effect at the end on your cancer, that's anti-initiating effect. Similarly, you give carcinogen and then you wait for a week and start a drug and look at the effect. So if you see the effect, well, the cells are initiated, so effect is going to be anti-promotional effect. So you have anti-initiating effect and you have anti-promoter. And we always keep one group where we start from -1 all the way through. So because sometimes what happens is that may have a effect, overall effect. It may not have selective effect like anti-initiator or anti-promoter. So you give it throughout your experiment, and then that's going to be a third group, right? And this is the one that we just kind of talked about. So there are many anti-initiators, or the initiator cells, many anti-promoters. This is like, this list even doesn't cover 1/100th of the list that's been tested, because numerous chemicals are tested. One of the problems in testing many potential cancer preventive agents in the in vivo system is expense. So where I come from, the institute has a huge big contract. It's called so-called master agreement. So we have master agreement with the National Cancer Institute, and only 2 or 3 labs in the country have it. So like mammary gland organ culture was me, and nobody else had it in the country. So anytime a National Cancer Institute wants to test a compound, they will send it to me. So every year they'll send me 25, 30 compounds to see if that has activity. The in vivo, I was not the principal investigator, but I was very heavily involved. That is also in our group for breast cancer and prostate cancer and urinary bladder and so on. So we tested many compounds, and some of the compounds I cannot read it through, so I'm going to read it through my frame here. And this has written like curcumin, EGCG, resveratrol, gingerol, just to name a few that have been tested. Now when we were working on retinoids with Michael Sporn, he was the one who was a retinoid person. So the idea was that instead of giving vitamin A, he isolated the retinoid compounds that are the analogs of retinoic acid. Now all trans retinoic acid, which is the principal component, there you have either retinol, which is good for your eyes, and then you have retinoic acid, which is good for considered as for cell proliferation. Now all trans retinoic acid is extremely toxic, so you cannot give all trans retinoic acid. And also there is a very fast conversion from all trans retinoic acid to 13-cis retinoic acid very rapidly in the body. So, and 13-cis retinoic acid is not active. So you have to make analogs of retinoid, and those were the ones that were tested in vivo, and they tested about 50 different analogs or more in vivo. And one that they found, which I'll show you, I'll show you. So this is just a concept of cancer prevention. The reason cancer prevention was very important at a time, or even now, is that cancer therapy, chemotherapy really works after the cancer has already occurred, whereas it takes about 10-15 years before the cancer actually becomes a cancer. So there are a lot of things that you can that's going on: cell proliferation, metabolism, signal transduction, cell cycle. Lots of things are going on in the process. So this was modified from De Flora, who is in Italy. He had put out this. I copied from his paper, and so there is a sea of carcinogen. There are many different things that you can do with compounds that can break any one or more of this system, and you may not get a cancer. That's why cancer prevention started getting a lot of importance. Now, before I go further, I want to say that which people, like physicians or clinics won't agree, or industries won't agree, is cancer prevention never became very popular in the clinics or in the industries. So you don't find too many pills or too many prescriptions, and the reason is that you are dealing with normal people. So to do a clinical trial, you need huge number of people, like thousands, 50,000 people. Whereas chemotherapy, you can get away using like phase 1 trial with 10 people or 15 people. Secondly, chemotherapy is expensive, and the industries can make huge amounts of money on chemotherapy. They cannot make money on chemoprevention. So that was the reason the chemotherapy has been given a lot of importance, but then the chemoprevention is a really good field to get into. So this is really from a chemopreventive agent here. I had generated this when I gave a talk at one time with vitamin D, and I replaced CPA for vitamin D. So what happens is there's few things that are important. The fundamental thing that's different between therapy and prevention is here you don't have a disease. So the effect of your drug or a compound has to be absolutely non-toxic, right? Because you're not going to get cancer. So why would you risk having toxicity? So there is it has to have no toxic effect, and you have food habit changes that you can do. You can start chemopreventive agents in a high risk group. So like for example, in case of breast cancer, it's hereditary, or if it's a BRCA positive people, parents are BRCA positive, or somebody in the family had a cancer. And I'm just saying breast cancer as a generic term because I worked on breast cancer, so it automatically just kind of flows. But that goes for many cancers that are hereditary. Okay, so in a high risk group, you can have the models which we talked about in earlier class, all the carcinogenesis models that are used for this thing. You can use mammary gland organ culture, we'll be talking about. You can do the cell transformation. The cell transformation is not as easy as it sounds. One of my students spent like 6 months transforming the MCF12F cells, which are normal mammary epithelial cells, with a carcinogen, and look at the effect of the compounds. We can do the colonic ACF, aberrant crypt foci, and we can do the mammary carcinogenesis studies like MNU, DMBA. Now in therapy, what happens is we have to look at the toxicity of the chemotherapeutic agent is very important, but there we look at risk versus benefit. So in other words, is the chemotherapeutic agent or treatment that we give to the patient, is that going to have a more benefit than the cost? So all chemotherapeutic agents or treatments cause side effects like nausea and vomiting and the headaches and hair fall off and all kinds of nasty side effects occur, but they are acceptable. The people accept it because the outcome is that you are going to have the cancer is going to be gone, or cancer inhibition is occurring. So that is risk versus benefit. For that, people use the models in the cancer therapy. So now we are going to use the cancer cells in the mouse. So mouse is going to reject the cancer human cell. So we have to use nude mice or athymic mice. So the immunity is compromised. There is no immunity. So all these studies that are done to evaluate the effect on the tumor growth or tumor cell growth, they all use athymic mice or nude mice. They are a little more expensive, but you can get by with very few animals. Usually people use like 4 or 5 animals a group in that. The reason is that the mice, each mouse is $50 to $60, and pretty expensive study. But that's what's used. And then here you have a high risk group of people. Here you actually have patients with cancer. So these are cancer patients. So in my case, I was talking at lunchtime. The problem that I was having is that now I'm trying to use cancer preventive agents as a cancer therapeutic agents. So one of the things I was telling yesterday, that we use cancer cells for our experiments. So anything that works on cancer cells is therapeutic in anybody's book. and we all use MCF7 and whatnot, the whole list of them. The second thing that I came up with this model is there is a area we really don't know in the human or in animals, that when prevention ends and therapy starts, right? So I call it a prevention of progression. We really don't know when there are few cells, like 1,000 cells, you cannot detect ductal carcinoma in situ, DCIS. We don't know whether it's there or not in the body. So that time I just said that, well, if you continue giving the preventive agent, you are also making it work on the early cancer cells. So I termed it as a prevention of progression, and this has been published also in the literature. So it's in my my CV. In some review articles I wrote, I also defined the term called prevention of progression. So you can use this compound throughout the cycle, either as prevention or as therapy. So the definition of chemopreventive agent, original definition was given by Michael Sporn in 1976, and so he said that any compound that inhibits or reverses the growth of a transformed cell is called chemopreventive. And in case the definition was modified by Wattenberg, Lee Wattenberg recently passed away. He's very old. But what he called is he divided them into several different parts, and he called it things that are like anti-initiators and anti-promoters, and he was working on colon cancer. And then in 2001, Michael Sporn came up with a new definition to introduce the molecular aspects to the cancer prevention. And then John Pasudo is a PI on this huge big project I was talking to you yesterday about. He's a dean of School of Pharmacy now in New York. And so we wrote a paper and we said that our chemopreventive agents are chemicals or combination of chemicals. Combination of chemicals was not involved in any other definitions, which can alter or reverse the expression or function of a molecular target. Because nowadays, in recent years, as I was telling, that molecular targets are important. What are the targets? Receptors are target, kinases are targets, adapter proteins are targets, signaling transducting molecules are targets. So the compounds that are specifically affecting to reverse or inhibit the growth of molecular targets that are responsible for transforming normal cell or supporting the proliferation of transformed cell. That's what we call chemoprevention. This is now very well accepted definition for chemoprevention. So the chemoprevention would use either natural or synthetic chemical agents to reverse or suppress and prevent carcinogenesis process. Now what happens is that original idea, or when chemoprevention word was introduced, at that time Sporn was using isolated compounds, and that went on for many years until even now that new compounds are being analyzed. Now the whole food concept is coming back, where there are other things in the foods. As I was telling you yesterday, I gave you example of turmeric, that the curcumin, which is very high concentration in turmeric, is not really absorbed by human cells or human cells, cancer cells. Now so Bharat Agarwal, he's a big guy in MD Anderson, recently retired, and he worked a lot on curcumin, and he was telling that, well, he started using the whole food now because there are enzymes in there that makes curcumin absorbable. So, and he showed the data on that. So I think that those are the reasons. You can do the target organs that we can do the test on is urinary bladder, breast, cervix, colorectal, ovary, head and neck, lung, prostate, skin, etc. These are natural synthetic compounds that people have worked on, are retinoids, big fields. Retinoids are vitamin A. Deltanoids is a term given by Michael Spohn, is called vitamin D related compounds. D. Those are called deltanoids. That term was coined by Michael Spohn, but it did not become as popular as retinoids. And then you have a phytochemicals, as we all, majority of us here are working on, are the new phytochemicals that are derived from new plants, edible or non-edible, and from fruits or from any other thing. And these are the non-steroidal anti-inflammatory agents called NSAIDs. They are used because one of the studies that you may have heard about aspirin. That aspirin a day is really good for heart, and also there was a study that was for colon. That the colon cancer was also, they say, take a pill a day. More recently, there was a study saying that aspirin a day would be good for breast cancer. Now I give an example what happens in terms of the overall picture of it. I had a study on aspirin in 1985, somewhere around 84, 85. So it was a beautiful study. So we had a low dose and high dose of aspirin, okay, and we gave it to the animals in breast cancer, mammary, carcinogenesis profile. We gave it till the animals died. The best study you can ever see. So that it's not like you cut off your study at 3 months or so nothing like that. Waited till every animal died of natural cause, and we showed that animals, the aspirin had no effect when we gave it a high dose, but aspirin had an effect when we gave it a low dose. Beautiful statistics and all that. So we talked to NCI, National Cancer Institute, because it was their money. So I said, we want to write this paper for Science or something like that at that time, or some really high-powered journal. They said no. He said, you must have done something wrong. Okay, so that there is no way that your low dose has better effect than your high dose. And 20 years later, everybody says that use low dose of aspirin and not the high dose. So that's the story of my life. What can you do? So here we go. So this we discussed yesterday. So I will talk about it, but I won't talk about it in too much detail, because remember, I'll say a little bit because she was not here. So I'll tell. So we have a chemopreventive agents. They could be either synthetic or natural products. It could be mixture, or it could be a pure chemopreventive agent. Now, the chemoprevention is either, it could be in population where we do the clinical trials, or we make the biomarker analysis for early diagnosis, or it could be experimental where you have a bunch of models, as we talked about, cell culture models, organ culture models, in vivo models, or we can do molecular chemoprevention. So we can say that, okay, the Ki60 is prevented, or you don't have the the P53 is is upregulated, or do the PCRs, RT-PCRs. Those are all molecular chemoprevention that we could do. And these are all, many of them are functional foods. There is a concept in the United States that many universities were forming a functional food program groups supported by the university. Our university had one at University of Illinois, and that's where lot of different people were involved, and good work was going on in there. So the fruits and food is very important in that regards. So as I had mentioned before, that this is from one of my very early papers, that in United States, as I was telling, that all these different plant products are available on the shelf. One of the reasons is that the FDA does not require approval for it to go on market. So you guys can mash up your drug and plant and make the peel of the extract and put it in the market, and FDA won't say anything. But they all have been tested with its property, like antioxidant property, or some lesions. Some of them, they're all different properties, and some of them are strong, some of them are very weak, depending on where they are published. Some are published in sort of not very high power journals, and then some of them have not been scrutinized as well. But you can go through each plant, what kind of activity that the plant has. You can go through that. I'm not going to go read it through. The couple of important ones that we are talking about is soy. You have a genistein in soy, which has been used quite a bit in cancer prevention. You have gingerol in ginger, which is also used in a great amount, and then there are lots of them here. And then here's some more. So like what I did here is that the compound, like you have curcumin in turmeric, or you have capsaicin in red peppers. So I had a paper on capsaicin in Journal of National Cancer Institute, which is has a huge impact factor, better than cancer research. So we worked on that and had a paper on that. Gingerol, all these I worked on. EGCG in green tea, genistein, soy, or they're all in here. Diallyl disulfide in garlic, and these are some of the common ones. Lycopene in tomato. So this is one comment on that for example, you really have to see, like for example, lycopene is present in tomato, but it's available, or it comes out only when tomato is cooked or is boiled. If you eat like salad, it ain't going to do anything for you. So you have to really remember where your compound is, or how far is going to work, or how well is going to come out in the solution. Okay, so in our drug discovery program, we had and I'll talk a little bit more later on, but then we go through the screening. So you use cancer cells, you use mouse mammary, gland organ culture, you screen. We have screened hundreds of compounds. So there was when my lab was very active for MMOC, we have done including the compound in the extract from here. So when our lab was doing active mouse mammary gland organ culture, I had practically done every compound that comes out in literature. You see the literature, somebody's paper, I said I've done this. So it was really good. Like we did hundreds and hundreds of them. And then what happens is you have a parent compound that that is present in the plant, or whatever plant derived. They synthesize the analogs, you screen the analogs, you select the active compound using these systems. Then you have to do the experimental carcinogenesis, you do molecular mechanisms of action, and then you do the preclinical toxicity, and then it can go to clinic. This step here is not done in the lab. They are done by the contract labs, because preclinical toxicity for a drug to do is a $500,000 project. So it's very expensive. Once it goes through, then you submit your folder to the FDA for getting its approval. Now this one I showed you yesterday because we were all getting bored with the videos. So I explained to you, but I'll go through one more time because ma'am wasn't here. Professor, I wasn't here yesterday. You weren't here, right, when I described the slide? Were you here? No? Okay. So we have a huge big program. Now it's no longer there, but it was for 15 years. And every year the National Cancer Institute gave $6, $7, $8 million a year to do that. It was a composite program with many PIs, many principal investigators in their own respect. Some, like we had the Duckinghorn, who is a editor-in-chief for Natural Products. We have Norm Farnsworth, who passed away now but is considered father of pharmacognosy. We have John Posito, who is a dean right now. He was a PI of the project. I was involved in it. Then you have Bob Moriarty, who worked with a couple of Nobel Prize winners. He's a synthetic chemist. And so this is a really big involvement, huge big project. Richard Moon was my boss, who is an in vivo carcinogenesis person. So what happens is that you select the plant from various countries, including India, spices, whatever. The plant, either we make the extract here, if it is India or Peru or wherever, because our resveratrol came from the grapes from Peru. So you make the extracts, you evaluate the extracts in 7 bioassays, and those bioassays are biochemically defined, not just cell proliferation. We look at the assays that have the steroid receptor, is the protein kinases, it has the activity in antioxidant activities, some sort of a Ki67 upregulation, things like that. So then if it is found positive, you have an active extract. That extract undergoes mouse mammary gland organ culture system. If it is active there, then it becomes a lead plant. If it is inactive in MMOC, it gets dropped and gets shelved. Now the lead plant goes through fractionation, and each plant is one thesis for a PhD student in a medicinal chemistry. So they fractionate them, and then all those fractions get evaluated in a selective bioassay. Selective bioassay is the one, that bioassay which was positive to show the activity in the extract. So only one assay is done here, and you select an active fraction, and then the medicinal chemists give that some name, that looking at the whatever they are doing with their NMRs and literature, and say, okay, you have a compound. Now many times the compound they find is already in the literature, so then they have to go back to the drawing board to see that, use another plant or see if there is another compound in there, because they have no interest in reinventing the wheels. They want to do something that's new, and that everybody does. PhD students, they don't want to work on something that's already been done. They want to do something very novel, which they can say, which they can claim their own. So then that gets involved. You have activity. Now you have possible chemopreventive agent. It goes through MMOC and ACF, aberrant crypt foci of colon. If it is active, it goes forward. If it's inactive, it gets dropped. So here, this is a judgment call. Say that, okay, no, it's not active in my system. So they'll say, okay, then we're going to drop the compound. That's it. Then if it is active, then it becomes active chemopreventive agent. Then it gets synthesized large amounts for in vivo carcinogenesis system against Richard Moon, which was also in our lab. And then you show the activity. If the activity comes up, you study the mechanism of action. You get new grants very easily, because now you have a new compound which was not known previously, has a very high activity, good potential, a right mechanism of actions with a huge number of preliminary data. So it gets reviewed well. It's you get a good review on that, and you got funded. So then we have group becomes larger, new students, new postdoctoral fellows, and that's how the whole system works. Okay. Now the synthesis, then it becomes, we have to synthesize it in a GMP, which is a good manufacturing practice, which is very expensive. You do that, you do preclinical toxicity, and then once it's approved by FDA, then you get the phase 1 clinical trials, and you have a chemopreventive agent. So as I was telling everybody yesterday, Professor, was that we are here, usually, we have to move forward. So the way to do it is to have the collaborations or whatever we do, but then we have to go to next step, identify a compound, something that is novel, that's not in the literature, and that will give you a really good recognition for the work we do. So what happens is, for example, several potentially active chemopreventive agents are obtained. You do it in cell culture, you identify lead compound, you determine activity, mechanism of action, you go in vivo, and then this is the protocol that I explained there. You have the primary test, you have a secondary test. If it's negative, you store it, you go forward if it is positive, you have active principle, you do secondary test. Secondary test is MMOC or ACF. It's negative, you drop it. If it's positive, you consider for development. So this slide is 2000, year 2002, 2003. At that time, we had done about 15,000 such tests, and we had done about 3,800 plants. And out of that, in short-term assays, we found 800 positive. About 800 compounds were positive, and the extracts, plant extracts. And these extracts evaluated in organ culture, out of 800, 229 were positive. So 500 plants were dropped at that stage. And then the active extract we evaluated in organ culture, and then we found only 70 positive from there, right here, which are very high activity. So we selected them, and then the active pure chemopreventive from the fractions, these are supposed to be fractions, that we tested, we found only 30 positive that are isolated chemopreventive agents. And we got some other from, I had other projects, I got some more, and then from this, based on toxicity and all that, we selected about 20 for further development. So the whole process, if you see, it's a very long, lengthy, time-consuming process, but it has a, it gives you a good feeling at the end if if you come out with something that's positive. Okay. So experimental models for that we use for the studies, as I said, our mammary gland organ culture, we did cell culture, toxicity, in vivo carcinogenesis, metabolism, and mechanism of action. So this is the protocol I use for the mammary gland organ culture. So just a step back in the mammary gland organ culture system, that was my PhD thesis. So the thesis paid off pretty well in my career. So the whole idea was, when I had a thesis where I started in Berkeley, was that my boss, who passed away a few months ago, Ranu Nandi, who was from Kolkata, and so he was the first one who had done the effect of hormones in vivo for mammary gland development. So in mouse, he would do the ovariectomy, he will do the pituitary removal, he will look at the adrenal removal, he do the double like ovary and pituitary removal, like lots of combinations. And he came up with the thing that, okay, if you remove this, the pituitary, then this happens to the mammary gland. If you remove ovary, the ducts, you don't get the lobules, you don't get the pregnancy-like structure. If you do this, this happens. So when I joined his lab, he says, let's do this in vitro. So I started doing the mammary gland organ culture. We took out the gland, put this in the culture, and I started putting hormones in there. And I came up with the hormonal combinations. So if you have insulin, prolactin, estrogen, progesterone, or insulin, prolactin, and aldosterone, those are three I like from many hormones and combinations. I showed that you can make the normal mammary gland from a ductal stage very early, from young animals. I can make it look like a gland that's in the pregnant mouse in 6 days. So you're cutting out the time, right? So it's a really good system. And then what I did is, you remove the hormones. So I said, okay, I let it go for a few days. I remove the hormones, it goes back to the normal state. So that's just like a normal mammary or breast physiology, that it goes back to the original stage. You can stimulate it again by putting hormones. And if you, while doing this, if you put hydrocortisone in the medium, and these are serum free medium, so there is no serum complication. Okay, I did it in serum free medium, and then you put a hydrocortisone in it, then that mammary gland secretes milk. In the culture dish, you can see the lipid droplets in the medium, and you can see the casein expression, you can see the alpha lactalbumin expression, which are the major proteins present in the milk. So then we took a step further, and I said, okay, during the development of this pregnancy stage, what happens if you put a carcinogen? So we dumped some DMBA in there. So if I put a DMBA, there's nothing happening. The glands look the same as you would see in pregnancy time, in about same time frame, 6-7 days. And then when I remove the hormones, then as the control, all the glands will go back to the ductal stage. Well, when I had a DMBA in, there were some patches, some areas where the cells did not regress. They remained. You can see the lumps, like you can see the growth of the cells that did not respond to the withdrawal of the hormones, meaning they had an altered hormonal responsiveness. Those cells were not responding to hormonal withdrawal, so I call them abnormal. So what we did, the next step is we took those cells and injected into the animals, and we had to do 2, 3, 4 generations, and then those lesions form tumors. Okay, so now that gives a great model. This all happens in 24 days. It's a 24-day culture. You have a gland growing for 10 days, third day you put DMBA, you remove the hormones for another 14 days, and you get these lesions. So if I have two groups, one group where I have control, where I put DMBA and go through this system, and the second group I have a drug in there, and if I don't see this growth, these lesions, then those are cancer preventive agents. Okay, that's the model. So here it's in descriptive form. In a 24-day culture, we give DMBA on day 3 for 24 hours, then remove it, and then you have growth promoting hormones, insulin, prolactin, aldosterone, or insulin, prolactin, estrogen, and progesterone, and I'll go to that in a second. And then you do the regression, you go up to 24 days. If you give the chemopreventive agent before the carcinogen, it's anti-initiator. If you give it afterwards, it's anti-promoter, and you keep it in the culture. Insulin, prolactin, aldosterone, hydrocortisone induce alveolar regions, whereas estrogen, progesterone, if I put, it gives you ductal lesions. Now in human, the cancers are all ductal lesions. So this is more relevant in terms of a human situation, whereas this one happens also, but this is more like a proliferative lesion. So if you look at the control in the absence of DMBA, when the regression occurs, all the regression has gone off, you go back to the ductal stage like this. However, in the presence of DMBA, the areas don't regress. There are areas that don't regress. And if you put vitamin D, for example, in this one at a 1 micromolar, this one looks like that. So that means it's a cancer preventive agent. It prevented the growth of those lesions in this particular. So this one we used as the first step for practically all the compounds that we isolated or identified, and then we go full blast on that and do many different things. So if you take a histopathological section in insulin, prolactin, aldosterone, these are the mammary alveolar lesions where you can see the alveoli, and they're very proliferative, very big thing. And if you look at it in the control, where you have the ductal ducts are all reversed, regressed, you will see like plain ducts. You will see some of this, but most of it is reversed. And if you have an active cancer preventive agent that you call is effective, the section will look like that. So now you're comparing that with this, right? We don't take sections every time. Only if you have to do immunohistochemistry or some parameters. Otherwise, we'll just go and my technician will just count the lesions or do the imaging and tell you whether there is any activity or there is, what percent, and then compare that with the control. So we will be counting some of that are not regressed. We count those also in the system. Now, if you have the estrogen, progesterone, and in the system instead of aldosterone, then you get ductal lesions. So if you look at the normal mammary gland after it's been regressed, the duct looks like that. This is the mammary duct. It's the epithelium. It is a lumen in there for the, the milk ducts or whatever you have in the animals, and then you have the epithelial cells like a single layer. Now what happens is that sometimes you get a hyperplastic. Remember hyperplasia? Number of cells increase. We see hyperplastic ducts. Okay, and sometimes this is dysplastic. Dysplasia is almost always going to cause cancer. So sometimes with DMBA we see a ductal lesion like this, and then you give the chemopreventive agent, then you have a big empty lumen here, and this is showing you the effect, because now here you have again a single cell layer like this with large lumen like here, and here you have the hyperplastic lesion with many layers of cells, and there is an intact lesion. So this is sort of pre-neoplastic, and this one is dysplastic. So this is bound to become cancer if you let it go long enough, and it occludes the duct. So now the duct has no lumen and is filled in with the all this abnormally growing cells. Now if you have a cancer therapy preventive agent in there, and then you can it's more like that. So then you can say that, well, this drug or this compound has a potential to be a cancer preventive agent. Make sense? Yes. Say yes is my thesis. Okay. So MMS is dead. In my lab, because I was the one who was doing it. My technician is still there, but my postdocs, they just moved on. They're doing their own little thing. Some other labs are doing it, but because I'm gone. So, and this was an expensive affair. Animals, maintaining incubators, and so no, there is a Mexican girl who worked for me for 15 years, lots of papers with me. Her name is Genoveva Murillo, and so she's running the projects that I have. Because what happened is, after I left, I still am involved. There are a couple of different projects that that got funded with good money, which I have going, but I don't have a lab. So it's like all it is, I go and do discussion. Somebody else has to finish the work. So you have I was explaining somebody earlier. In terms of chemoprevention of mammary carcinogenesis, you have to have a maximum dose, tolerated dose. As I explained in the previous lecture, that you have to take, I think there was a question, and then I answered saying that you have to have your drug into 6 different concentrations and find the concentration that's toxic, step down one concentration, and then take 80% of that to look at the effect of that drug in mammary carcinogenesis. If it is effective, then you have a chemopreventive agent. You can test it with the MNU or DMBA induced carcinogenesis, and then you look at the initiation, promotion, progression model, and you study the toxicity. The protocol for that is very similar. We take a 35-day-old animal, and for anti-initiation study, we start them on the compound, and we overlap the carcinogen. We give carcinogen on day 50. MNU is given intravenously in the clavicle and near your neck. DMBA is giving gavage through the gastric feeding, and then you just let it go. And for the anti-promotional agents, we give the compound from here to here. And then for screening, we go all the way from here to the end of the study. And the study usually goes for 180 days for this. For screening, the study goes only up to 100 days. So these are some of the results. I'll show this slide again when I talk about vitamin D later, day after tomorrow. But if you do the project like this with the MNU, you have a control incidence of tumors, and then you have a inhibition of carcinogenesis, and then at the intermediate dose you have a intermediate inhibition. And then if you look at this, one of the things that if you have done the pre-screening of dose tolerance, then you know that the effect you are getting is non-toxic, because you already taken out the toxicity out of the picture, right? And the way you measure whether there was a toxicity or not is you measure body weights. So you weigh the animals, and you always give a picture of body weights when you give your picture of incidence, because if there was a decrease in body weight like this, then these results don't mean anything, because that means that it's because you had a loss of body weight, the tumors were starving, and you got the negative or the positive effect. Okay? And the bioactive compounds that affects all the system is you have a carcinogen metabolism occurring, cell cycle involved, DNA repair, cell differentiation, hormonal regulation, and cell apoptosis. So here is an example from the plant. So for example, when we had the plant, there's only one or two slides I put just because you are also in similar area. So when the plant extracts come to our lab, they have a P number. Okay, this is a plant number. And then all this plant number, the compounds were derived. So these are all the different compounds that have been derived from these plants, and they have been tested. So this is just an example I gave. So for example, in this plant here, the medicinal chemist had identified a compound called carpolacton A. Now this plant, as I was telling you yesterday, is a tomatillo from where we make salsa. So it's an edible plant, and then you got the compound called the ixocarpolacton. Catechins, some of them have been worked on pretty extensively. Some of them have been dropped. So this is an example to the narrow it. Then you narrow it down. So for example, in this particular plant, you came up with a compound called texcarol. I don't remember that compound because it must not have very good activity. Then you have this one. You come up with the odorine. This was taken a little bit farther down. But then the ixocarpolacton, one of my students, who was that's really another thing. The students, how they become interested in research. Julie Choi is this girl who got direct admission after high school. Here everybody gets direct admission in medicine. In the United States, you get after 4 years of college. So you go through 12 years of high school, then you have 4 years of college, then you get into medical school, and you do 5 more years. So some students, they have a program called GPPA. That means guaranteed admission into medical school after high school. They're very bright kids. So Julie was a GPPA. She came to our lab, and I asked her to work up this ixocarpolacton project, and she was really getting involved and all that. And then she got admission into medicine. She was doing her, she started doing her medical rotation. She came back and she said, I want to do PhD with you, because she says that this medicine is not really going to be, my cup of tea. Well, she's practicing medicine now, but she did get PhD, so she's MD PhD now. But she worked on this ixacarpolactone, but her thesis was not on ixacarpolactone. She worked on a compound called azurine, which is from bacteria, and then she had a little 28 amino acid peptide that she isolated and worked on that for melanoma, and she got her PhD in that. So this ixacarpolactone, we saw that it was very active. These are the colon cancer cell lines, and showed the activity in those. And so once it shows activity in one of these cell types, then we take it further, and then it becomes a targeted compound for colon cancer. And then it goes forward. So this is the structure of ixacarpolactone. I showed this. I put this to show how the structures look like. This is a flow cytometry, and if you look at it, the ixacarpolactone has a G2 arrest. The cells are getting arrested in G2, and then this is the breakdown, and you can, not necessary to see right now, otherwise you won't be able to make it. So it's a G2 arrest. If you look at the its effect on apoptosis, and yesterday I told you that you have to look at apoptosis at several different ways, and this is control with rounded nice green cells, normal. When you look at the apoptotic cell, you see this blabbing. See this blabbing, like bubble type of things that come up in the cell. That means the cell is undergoing apoptosis. Okay, so that's what you see. When you see the orange structure like this, like that means the cell is dying. And so you look for this blabbing. So that's the cell apoptosis, and we showed that the cell apoptosis is occurring, and you count the number of cells in a field of your microscope and see how many cells are undergoing apoptosis, how many are dead. The dead cells are going to tell you how toxic it is, and normal cells are going to say that which cells are not affected by it. There is another nuclear fragmentation. So you can see the shrinkage and of the interior, like condensation of all the chromatin that's occurring in the ixacarpolactone treated animals, cells. There's some more. I won't go into BIMBOD because the apoptosis. So what I'm showing all this to show that what the logic occurs. So you said, okay, selected ixacarpolactone, showed the inhibition, then you ran the apoptosis and say, okay, well, it's getting apoptosis. So then you have to do 2-3 different ways. The one procedure is okay, but when you say confirm it by 2 or 3 different procedures, then it's really good. So you confirm that it's apoptosis. Then what are the molecules in apoptosis, like molecular markers that you want to see? So you want to look at the BCL2, BAX, BAD, and she selected the one based on the domains that are BH2, BH1, these 4 domains. This is a BCL2 gene structure with 4 BH domains, where there is a BIMBOD that was very new at that time where she was doing. BIMBOD does not have the later two structural domains of the BCL2 gene, and that was really correlated with the cell apoptosis. And she showed that where the BIMBOD was actually, you can look at the control and ixacarpolactone with different days, and you can see as you go that there was an enhancement in that, and BCL2 had not that much of an effect, because that was like still normalized with your actin. Now the colon cancer model for ACF, we use the azoxymethane 2 injection as a model. So what happens is that you take the animals and then you give them 2 injections of AOM, and you start your treatment for promotion, just same protocol as mammary. Initiation here, promotion there, You sacrifice the animal standards after a week after that, and then you can study the aberrant crypt foci. And for carcinogenesis study, you take it further down. So it's anti-initiation, anti-promotion, and the entire time frame to study the activity. And so these are the crypts. Okay, these are the crypts. So either the colon have no crypts, 1 or 2 crypts, like here, or it has more than, like it has 3 crypts, 3 here, or it has 4 crypts, or it has more than 6 and more. So these are all aberrant crypt foci. We have them in the human also, and that's used for human as a marker for cancer prevention also in many systems. Now, while doing that, we also identified an enzyme called hexosaminidase. So hexosaminidase is a normal constituent of a normal cell. So this is normal, is present when you have the loss of hexosaminidase. You will see the green structure like that. Okay, so this is the LOH of normal area. This is the LOH of the ACF, loss of LOH, and then you can correlate this with the number of crypts, and that you can use as a marker. And I won't go into this, but this slide shows the different compounds. And we had like papers. We have daguline paper, we have saponin paper, we have pomegranate paper, and we have different flower papers showing the effect on ACF. And this, we talked about it yesterday, is how the wind signaling works. And let me see my time. 5 minutes? 5 minutes? Or call it off? Do it in the afternoon? Lunch? Okay. Let's break for lunch. Alright. So what I'll do is I'll finish off this talk. There are 5-10 slides left, not too many. And then in the afternoon we'll be talking about work that's being done in the lab. So this is a little more interesting than listening to the videos. You want me to run it? Is it okay? Oh, I thought that, okay. So, yeah, so daguline, I'm going to be talking a lot about daguline later on, because that's my newfound drug that we have. It was all in the Chicago Tribune and newspapers and everything. UIC means University of Illinois, Chicago. Attacks cancer's root. I mean, whatever. Okay, so we have worked on a variety of different compounds. We have worked on vitamin D. We have worked on the sapote, means chiku, right? So sapote, Mondalia cerevisiae, this is a daguline. Sapotin comes from this. We worked on resveratrol. We worked on pomegranate, and we worked on isocarpolactone. This is just the nutritional, like fruit based. And we worked on the carcinogens in effect of resveratrol on cigarette smoke induced lung cancers, and I'll talk about it also later on. This is a really important study, but it will take me a little bit more time to explain this. But this was published. Lorna White was my postdoc from Ireland, and she did this work. It's really beautiful work, but I can explain it later on. It's a new concept in there. And the TRAMP model that many of my friends are using in USA, it induces the prostate cancer in the animals in natural form. So these ones are some of the results we showed. This is a resveratrol we published in Science, showing you the activity of resveratrol that's present in red wine and in grapes. That first paper was from our lab. The betulinic acid, I was not there, but Pazzuto showed that he published it in Nature Medicine. Then this one is for bromoflavone. This is dropped now because of the toxicity found later on and having no effect. But all you can see is that if you look at the control versus inhibition, they have a very dramatic effects, and that's why I'm showing. So that makes the case for these compounds for further development. So any of these compounds that you pick up and see the literature, you can pick up and make your own project on that, and it works very well. This is Dagulin. I'll be talking about Dagulin later on, but this is a cancer research, but the original Dagulin paper came out in Nature Medicine. And this one here is, this is Dagulin. So the one I was showing you, there are two different concentrations. You had just dramatic effect on the skin carcinogenesis. And some of the other compounds that we have papers on and we worked on are brasinin from cabbage, resveratrol from grape, brucitrol from non-edible plant, sapotin from chiku, ixocarpolactone from tomato. And there are lots of other chemopreventive agents that, and this slide you have, so you can go through these in your system. Now, few more minutes. No? Chalo. Yeah, okay. This is drug development, how the drug is going through the FDA. I'll talk about it in the afternoon.
Very elaborate overview of all the chemopreventive and chemotherapeutic agents. It has in mind that it must not overdose you. The thing should be absorbed.
Yeah.
At the age of 70, he is since morning he is doing all that.
It was really fortunate to
I
meet.
think in 2000, I went for a conference in Switzerland, and there he delivered lecture on, I think, vitamin D, and I listened to him. He was the only Indian, I think, there, and I was there. And I was with my family, my small son of 1.5 years old.
We went to Alps.
And then we went for sightseeing and we had interaction.
Yes.
And we were also working on the same line. My first student, Kamalji, she did good work. And I was carrying the extracts with me that somebody should analyze those extracts, because we were at very infancy stage of work. We were not having much funds to establish the different assays. So he was very kind to have my extracts, and I told him that if you check these extracts for MMOC assay, which he has explained to you,